Read the package insert carefully before use. The kit is composed of the test cassette, buffer, and dropper. Allow the test materials and specimen to reach room temperature before testing. Remove the test cassette from the sealed foil pouch. Do not open the pouch unless you are ready to begin the test. Place the test cassette on a dry, flat surface. When using a disposable dropper for serum or plasma specimen, draw specimen until it reaches the specimen line. Hold the dropper vertically and transfer 1 drop of drawn specimen into the sample well labeled as S on the test cassette. Then add 2 drops of buffer to the buffer well, labeled as B immediately after. For whole blood specimen, draw the specimen into the dropper. Hold the dropper vertically and transfer 1 drop of whole blood to the sample well. Then add 2 drops of buffer to the buffer well immediately after. Start the timer. If after 2 minutes the sample has not moved across the test window or if blood is still present in the sample well, add 1 additional drop of buffer to the buffer well. Read the test result in 10 minutes. Do not interpret the results after 15 minutes. IgG positive. 2 red lines appear on both C and G regions. IgM positive. 2 red lines appear on both C and M regions. IgG and IgM positive. All 3 red lines appear on C, G, and M regions. Negative. Only 1 red line appears in the control region. Invalid. The control line remains completely or partially blue. The control line must completely change from blue to red. Review the procedure and repeat the test with a new test cassette if an invalid result appears. Warning: this product is intended for professional use only. Strictly follow the directions for use within the package insert. In general, antibodies can be detected 1 to 3 weeks after infection. Results should be used in conjunction with other data such as symptoms, results from other tests, and clinical observations.